Hi guys, this is Kumar. In this podcast, I'm going to go over the process of transcription. As you know, protein synthesis is having two steps. The first step is transcription and the second step is translation. So in this video, I'm going to go over the process of transcription. You are aware of the structure of DNA. DNA is having two strands and the nitrogenous bases, which are the codes for the protein synthesis. So DNA uses the messenger RNA to make protein. So the process of making RNA from the codes of DNA, that process is called as transcription, or the making of the RNA from DNA. That is, the RNA gets the code from DNA, and that process of making RNA from DNA is called as transcription. And when RNA uses these codes from DNA to transform into protein, that process is called as translation. So in this video, I'm going to discuss the process of transcription. For that, you need to know the structure of RNA. So how RNA is different from DNA. RNA is having a different sugar from DNA, which is called as ribose sugar, and DNA was having deoxyribose. DNA is a single strand, but DNA is a double strand and RNA is a single strand. And RNA has a different base. So if you remember, DNA was having 4 bases. One was adenine, thymine, guanine, and cytosine. But in RNA, there is no thymine. And instead of thymine, there is uracil. So in this video, I'm going to mention one vocabulary word, which is a messenger RNA. So what is a messenger RNA? Messenger RNA carries the nucleotide sequence from nucleus to ribosome. So there are 2 other RNA which I will be mentioning in the second step of protein synthesis, that is translation, and those 2 RNAs are going to be transfer RNA and ribosomal RNA. This is the structure of DNA, which is having 2 strands, strand 1 and strand 2. It is having different codes in the form of nitrogenous bases for protein synthesis. And these codes are guanine, adenosine, cytosine, and thymine. One strand of DNA runs from 5' to 3', and the other complementary strand runs from 3' to 5', and that's why it is called as anti-parallel, because one strand of DNA runs from 5' to 3', and the second strand runs from 3' to 5'. So these 2 strands of DNA are having the different sequence of the nitrogenous bases in the form of guanine, adenosine, cytosine, and thymine. So guanine pairs with cytosine and adenine pairs with thymine. On DNA, there are 2 regions for the process of transcription. The region where the transcription start is called as the promoter region, and the region where the process of transcription stop is called as the terminator region. So you can remember, promoter is the region where the transcription or the process of transcription starts, and terminator is the region where the process of transcription or the making of RNA stops. So there is another important structure which helps in the transcription or making of RNA, and that is RNA polymerase. This structure or the enzyme is really important in making of the messenger RNA, or taking the codes from DNA to make an RNA. what RNA polymerase does is it binds to the promoter region and moves down the DNA strands. So as the RNA polymerase moves down the DNA strand, it makes the RNA strands from the codes or the nitrogenous bases, and it takes the codes complementary to the mRNA from the DNA strands. And the strands which it takes the codes is called as the template strand. So among the two DNA strands, polymerase uses one strand to read the bases to make RNA, and that strand is called as the template strand. The other strand which RNA polymerase do not use is called as the gene strand. As the RNA polymerase moves down the DNA template, it keeps on adding the nitrogenous bases complementary to the DNA strand. So remember, the adenine always is going to have uracil on the messenger RNA. So once the RNA polymerase reaches the terminator region, it unwinds the DNA strands. And now you have got a pre-mRNA. So why we call that as a pre-mRNA? Because we cannot use this messenger RNA for protein synthesis. It needs to be modified, and that process of modifying this pre-messenger RNA is called as splicing. So how it is modified? There are some enzymes which can degrade the pre-mRNA. So for that, the messenger RNA attaches a 5' cap to its 5' region, and to the other end it attaches a poly A cap to that region. If you look at the messenger RNA or the pre-messenger RNA, it has got two different regions. So one region which is called as exon, and on exon are the codes which can be used for protein synthesis. And there are some regions which are called as introns, and introns are having the codes which cannot be used for the protein synthesis, or they are the non-coding sequence. So we need to get rid of these regions which are not going to be used in protein synthesis. So how we are going to do that? So there is a structure which we can take help is the spliceosome. So what spliceosome does is it attaches to the region of introns, and it cuts the introns by binding to the intron region, and it cuts out, and what are left are the exons. So now when the regions which are left on the mRNA are only the exons which are going to be used for protein synthesis. Now this mRNA is ready for protein synthesis. So we are going to look into the next step of protein synthesis, that is translation.